Hello, everybody. Thanks for joining us. Welcome from snowy Boston. Our speaker today is Huaiying Zhang from Carnegie Mellon. We're really glad that she's agreed to do this lecture because she's really been in the condensate field for quite a long time. She did work previously both in Cliff's lab and Amy's lab. And for example, she was a co-author on that really important 2015 molecular cell paper with both Amy and Cliff on polyglutamine interactions with RNA. It's just one example of, of many. And she's also, of course, working more recently these days in cancer, particularly in this old but still incredibly important topic of telomeres. And she's already reported on quite a lot of that work, but she'll be talking about more of that today. And she's really remarkably diverse in the tools that she uses. To call her work multidisciplinary, I think would barely cover it. She's really fearless about applying diverse imaging and biophysical methods to her work. And she's also been involved in helping to develop some really creative chemical tools as well that she's also reported on. And so today she'll be telling us about her work on liquid-liquid phase separation in telomere maintenance in ALT cancers. So thank you so much for giving this lecture. We're looking forward to it.
Thank you, Mark, for those kind words. I'm super excited to be here today. Next slide, please. We're having some technical issues, so you're going to hear me a lot, say a lot. Next. Yeah, I'm very excited to be here talking about our work in cancer and phase separation. Next, please. So as a chemical engineer by training, my first memory about phase separation was actually from my polymer class, where we learn from studies like this, where researchers look into droplets formed by polymer solution, and with map the phase diagrams, looking at how phase behavior is affected by temperature, the polymer chain length, etc. And next, please. So when I transitioned into biology from a postdoc, I was fascinated to find out that this phenomenon is actually used in the cell. And on the left, you can see Cliff's pioneer work looking at the liquid behavior of P granules and look at how they drip, how they fuse. This led to the whole field of phase separation as an organizer for biochemistry. On the right, you can see the beautiful droplets reconstituted by the Rosen lab, which reviewed the multivalency as the driving force for phase separation. Next, please. And you can imagine how excited I was when I was joining the Summer Institute as a postdoc with Amy Gladfelder to work on protein phase separation. It's just like all coming around. So next, please. So the protein we're working on is a polyQ containing protein, as Mark was referring to. It is an RNA binding protein. It has RNA recognition motif on the at the N-terminus, and it has this polyQ stretch in the middle. Next, please. And this polyQ domain is highly disordered, as you can see in this prediction here. And next, please. So when we purify this protein, we see nice droplet formation. Interestingly, the formation of the droplets depends on the polyQ domain. However, the polyQ domain is not sufficient to drive phase separation of this protein alone. Next, please. And then we look at how the RNA binding affects the phase behavior at physiological conditions, meaning at 150 millimolar salt, at nanomolar protein concentrations. The protein alone does not phase separate. However, when we add the RNA that can be specifically interacting with this protein, we see droplet formation. And we don't see the droplet formation when we add non-specific RNA, meaning total RNA, to normalize the charge. This indicates that it is the specific interaction between the RNA and the protein that can shift the phase boundary of the protein. Next, please. And then I went to Cliff's lab, where we use microbiology to look at the material property of the droplets. Here you can see here in the droplets, we use the red are the beads that we embed in the droplet, and then the green is the protein that is labeled, and the RNA is not labeled in this case. And then we track the movement of those beads. and then we plot their displacement over time. Next, please. As in this MMSD versus time curve. Next, please. And then you can see as RNA concentration increases from the green to from the red to the green, you can see that the beads move slower, meaning that the droplets is become more viscous. And indeed, next please. And when we get the viscosity from the movement of the beads, and we can see that as the RNA concentration increases, the viscosity of the droplet increases. So this is a very nice tools will allow us to determine what's driving and what are the domains of the protein and what the interactions that can drive phase separation, and how the material property is affected by different components in the droplets. Next, please. So when I started my lab at Carnegie Mellon University, I really want to look into what is the relevance of phase separation and how does that to do with cell function and with diseases. In particular, we're interested in looking into how does phase separation can be linked to cancer, how do cancer cells use this process, or what we run for several liquid phase separation process. Next, please. So I'm going to share with you our work on the particular cancer cells that use a specific way to maintain the telomere length. All cancer cells have to actively maintain the telomere length. And this is because the telomeres will be shortened with every cell division because the replication machinery cannot duplicate all the way to the ends of the chromosome. Therefore, every time cell divides, the telomere gets shortened. And when telomeres become too short, it triggers a cell cycle arrest. Therefore, for all cancer cells to keep dividing, they have to actively maintain the telomere length. Next, please. Majority of cells reactivate telomerase, which is a reverse transcriptase that use RNA as a template to add back the lost telomere DNA. However, there's a significant amount of cancer cells actually do not reactive telomerase. What they use is called a alternative lengthening telomere pathway. We call it ALT pathway. This is a pathway that is based on homology-directed DNA repair, meaning that one telomere uses another telomere as a template to repair, and the detailed molecular mechanisms are still under investigation. As you can see from this chromosome spread, one feature of all those cancer cells is that the telomere length is very heterogeneous. You can see the pink, which stands for the telomere, is very heterogeneous in those cancer cells compared to a non-ALT cancer cells, for example. And interestingly, for cancer cells that are telomerase positive, next please, if they are subject to telomerase inhibitor, some of them will die, but some of them actually will start to adopt this ALT pathway for telomere maintenance. And this becomes an increasing problem for cancer therapy using telomerase inhibitor. Next, please. So in addition to this heterogeneity in telomere length, there are many other unique features of those ALT cancer cells that are used for diagnosis, because unlike telomerase positive cancer cells, ALT cancer cells do not have a molecule specifically that can be used as a unique marker. Therefore, it uses a combination of several phenotypes. And one of them is the relocation of the PML body to the telomeres. PML bodies are membranous organelles that exist in the nucleus of normal cells, and the number and size change depending on the cell cycle, depending on the status of the cell, such as stress condition. And however, only in those cancer cells, the PML bodies are found to localize to the telomere. As you can see here, is showing you the image of some cancer tissue. The PML body that's stained by the PML protein localized perfectly with the telomere DNA. Interestingly, the telomeres actually cluster within ABBs, meaning that they show as big punctile, as shown here in this red staining of the telomere DNA. And this clustering is thought to provide template, bring telomeres together so one can use another as repair template for homology-directed DNA repair. So those are used in diagnosis. Then they got people ask, can we use those phenotypes for cancer therapy then? And this is hindered by our, next please, There's a lack of understanding how actually do ABBs form, meaning how do those PMO bodies relocalize to the telomeres? Are they actually nucleated from the telomeres, or they actually are the existing PMO body but somehow be relocalized to the telomeres? And then how do they contribute to the cancer pathway? Next, please. So the Roger Grunberg at Penn, they developed an assay by introducing DNA damage on the telomere. They find out that this damage response on the telomere can lead to ABB formation, telomere clustering within ABB, and other phenotypes of the OD pathway, including telomere elongation. And so when I was collaborating with them as a postdoc in Michael Lampson's lab, and I looked at these movies of telomere clustering, and I thought, oh, they just look like droplet fusion. So showing here on the bottom left, you can see this process of the reduction in the aspect ratio where the ABBs actually round up, and if we swap them, we will see the dynamic exchange of component. Next, please. This led me to hypothesize, next, please, that those might be actually ABB bodies formed as a response of DNA damage on those telomeres. And then ABB as a condensate with this liquid behavior, next please, that will allow them to fuse. And this fusion process then will allow telomere cluster within the ABBs. Next please. And moreover, this concentration process can provide a unique opportunity for the ABBs actually to selectively enrich DNA repair factors on the telomeres and to allow DNA repair to happen. What's unique about telomeres, it is for human telomere, is that it is a linear end, so that the telomere has specific structures and properties prevent the cells from recognizing as a damage site. Therefore, actually, the active mechanisms are placed to prevent DNA repair factors to be localized to the telomeres. Therefore, the formation of ABB can provide this unique chemical environment that actually allows the DNA repair factors to be enriched. Next, please. And then after clustering telomeres together and the enrichment DNA repair factor, the ABB then can be a place where allows telomere elongation to happen. Next, please. So to test this hypothesis, we ask ourselves, how do we test this? Because DNA damage response is a really complex process. In addition to ABB formation, there are many other things going on. For example, there are DNA damage protein formation, and that some of them are not proteins, are not part of the ABB process. And then how do we decouple the contribution of those from the ABB then? Then our approach was to try to induce de novo ABBs without DNA damage. That way we can specifically decouple this function from ABB formation from other aspects of DNA damage response. Next, please. So one tool that would allow us to do so is opgenetics, which use light to control genetically coded light-sensitive proteins. Next, please. And these tools are widely used in a lot of cellular processes, particularly those dynamic cellular processes where spatial temporal organization is essential, such as phase separation. Because the laser, by using laser, we can have really good spatial and temporal control of where we want things to happen. And then today I'm going to share with you some of the tools we developed. Instead of using light-sensitive proteins, we create small molecules that are light-sensitive, which provide some unique properties that allow us to control phase separation in cells. Next, please. So those tools are based on a modular design of photo-caged chemical dimerizer, where we choose two small molecules that can each interact with the protein. Next, please. The red pair is a small molecule called TMP that can interact with the bacteria, EDHFR, and this interaction is non-covalent. Next, please. On the right, the green is a halo tag system where the halo ligand binds to the halo enzyme, and this is a covalent binding. Next, please. And then we put a small molecule, we put the linker between those two small molecules, the TMP and the halo, that becomes the dimerizer. Next, please. To make this photosensitive, what we did was we put a photo cage on the TMP so that this dimerization is prevented unless we use a light to get rid of the photo cage. Next, please. And then the photo cage we use is NVOC. Next, please. And this is the structure of the dimerizer where we call it TNH for NVOC TMP halo. And this system is unique compared to other system, optogenetic system. It's based on heterodimerization, meaning that we can dimerize two different proteins together. And also we particularly choose the halo tag system to be covalent and the TMP to be non-covalent so that we can use actually the halo to localize, to anchor the dimerizers to a subcellular localization, therefore would allow us to recruit proteins specifically to a subcellular localization. Next, please. I'm going to show you an example how we use this tool to recruit a protein, in this case, just a model protein of M. cherry to centromeres, which is labeled in green where we fuse the halo to a centromere protein. As you see here, in the green is the centromere, and then the red is the M. cherry, which is diffusively localized in the cell. And then if we use a white field, just a whole field of UV to activate the dimerizer to get rid of that photo cage, and to turn on the dimerization. Next, please. You will see that M. cherry now is recruited to where the centromeres are. Next, please. Then we can also achieve spatial control in that we only want to recruit to one centromere. The advantage of doing that is we can use other centromeres as the internal control to look at the behavior of that specific centromere. Next, please. So here, showing here is again the centromere, and in this case we use a 4-5 laser to specifically target one centromere. Next, please. As you can see before the laser, and you don't see centromere there. Next, please. And once we turn on the laser, and now you can see recruitment in a couple of seconds, and then the M. cherry is recruited to only one of the centromeres. Next, please. And we expand the toolbox to include other functionality by shifting things around. This is the advantage, again, of this modular design. You've seen the NTH, which is a caged version. Next. And if we move the NVOC in the middle, now it becomes an uncaged, cleavable version, meaning that you can add the dimerizer, proteins will be recruited to where the subcellular locations you want, but then you can use light to reverse the dimerization and then release protein from that localization and then follow its functionality. Next, please. We also created a version where we use a different photo cage. This uses a different wavelength and to uncage the dimerizer. Next, please. And this allows us to combine them together because they're orthogonal. The NVOC is activated at 4-5 laser, and then the coumarin is activated at 444 laser. So now we have a caged and a cleavable dimerizer where we can use 4-5, where we can use actually the coumarin is on the cage, so we can use 444 to control the recruitment, and then we can use the 4-5 to cleave the molecule and then release the protein. So we've created a library of those dimerizers, and then another unique property of those dimerizers is you can have the same engineered cell background, but by switching dimerizers allows you to have different functionality. For example, you can use the caged dimerizer, uncaged dimerizer just with the TMP halo. You can add it to a population of cells, do biochemistry, etc. And also once the cage is cleaved, and then the dimerization is persisted through a long time, so you can use long-term imaging without using the light to keep the dimerization on. So we use those to study several cellular process, including organelle control, kinetical function. Next, please. And then in this case, then we want to, in my lab, we want to adopt these tools to study ABB function, to induce ABB without DNA damage. So what we want to do is to recruit a protein to the telomeres, and then this protein will lead to ABB formation without DNA damage. Then we ask, what kind of protein do we recruit then? Next, please. Then this requires us to ask what could be the driving force for ABB condensation. Next, please. And then we went to literature, we find out another unique feature for those cancer cells is somehow the telomere proteins are uniquely modified by small ubiquitin-like modify. And this stimulation process, next please, is required for the ABB formation. Next please. Also, we look into the components in the ABB condensate, and then what we find out is a lot of the components either have stimulation sites or they contain SIM, which is a small motif that can non-specifically interact with SUMO. Next, please. And the Mark Rosenab has already shown that the multivalent interaction, this non-covalent interaction between SUMO and SIM can lead to phase separation. And therefore, our hypothesis became, next, please, that the ABB formation is fundamentally triggered by a telomere stimulation. This SUMO-SIM interaction then drives ABB condensation. Next, please. So in agreement with this, we look at whether actually SUMO is enriched on the telomere after we induce DNA damage on the telomere, which leads to ABB formation. So in this case, we use the Roger Grunberg assay where we use the nucleus fork1 to induce DNA damage on the telomere. And here, in this case, the fork1 is tethered, it's fused to the telomere binding protein TF1. So what you're seeing fork1 is where the telomeres are. And you can see the SUMO, which is the green, localized perfectly with the fork1 telomere signal, meaning that we see enrichment of SUMO on the telomere after DNA damage. Next, please. And this is not seen when we use a fork1 mutant that is enzymatically dead, meaning that it is the DNA damage after DNA damage response, the SUMO is enriched on the telomere. Next, please. I've just shown you the quantification where you can look at the percent of telomeres with SUMO foci. We can also look at intensity, and both we see a significant difference between those two cases. Again, meaning that the telomeres are stimulated after DNA damage response. Next, please. So then this leads to the hypothesis where the DNA damage response that trigger ABB formation is the stimulation process. So after telomere proteins are stimulated, then it can recruit proteins that contain SUMO-SIM, which may include PML, which has 3 stimulation sites and 1 SIM. So after the enrichment of SUMO-SIM on the telomere, then the local enrichment of SUMO-SIM leads to the multivalent interaction between SUMO-SIM, then leads to phase separation and the formation of ABB. Next, please. So we want to use our optionality tools to mimic this simulation process without DNA damage. And then what we want to do, instead of recruiting SUMO, we want to recruit SIM instead, because we don't want to overexpress SUMO, which is involved in a lot of processes. And so the idea is if we recruit SIM to the telomere, then the SIM will then bring the endogenous SUMO there to mimic the stimulation process, and they recruit other proteins containing SUMO-SIM, then leads to phase separation. Next, please. And to do that, we fuse the SIM to the EDHFR and cherry for visualization, and then we fuse the halo to the telomere binding protein TF1 so that the dimerizer is anchored on the telomere. And then we first use immunofluorescence to see if we recruit SIM, can we actually enrich SUMO like we hypothesized. Indeed, as shown here in this, you can see perfect collateralization between SIM and the telomere and the SUMO foci. Next, please. And this is with wild type SIM. However, if we use a SIM mutant that cannot interact with SUMO, we don't see such enrichment. And so meaning that it is the specific interaction between SUMO and SIM that is enriching SUMO to the telomere after dimerization. Next, please. So then we use live imaging to visualize this process, what happens once we recruit SIM to the telomeres. And this is before dimerization, and the SIM is diffusive on the left, and then the telomere is in the middle, which is in the green in the merged image. And when we turn on the dimerization, next please. And you can see the foci start to round up, and then they bump into each other and they fuse, and you see here in that square that I highlighted for you. And so this kind of indicates that there is phase separation going on, and there's also droplet fusion. Next, please. Next, please. Okay, good. So this is the snapshot of some, and you can see the SIM is recruited initially and then starts to round up, and you can see actually halo TF1 channel actually also start to round up as well. And this is because the TF1 that after we add the dimerizers, the TF1 is actually linked to the physically linked to the SIM. So when SIM undergoes phase separation, then the TF1 also undergoes phase separation with SIM. So it behaves just like the SIM. Next, please. And then when we quantify the intensity of foci per foci, the integral intensity, which indicates the size and also the intensity, the brightness of the foci, and you can see that SIM, which is in magenta, start to increase because of recruitment and also because of phase separation. And you can see the TF1, which is the green, also start to increase as SIM is recruited. As I said, this is because it's linked to the SIM. So when SIM undergoes phase separation, it starts to enrich extra other than the ones that bind to the telomere DNA. But also this is because telomeres start to cluster, so therefore they become bigger. As you can see here on the right, where I quantified the number of foci over time, you can see they start to decrease after SIM is recruited, after the phase separation takes place. This is because of telomere clustering. Next, please. And showing you an event of this fusion event, which is also telomere clustering, because we're looking at the telomeres, and you can see the rounding and the reduction of aspect ratio to 1, and also dynamic exchange of components by using FAB, agreeing with that this is a liquid condensate that we are forming. Next, please. And then we look at what happens when we recruit the SIM that cannot interact with SUMO. I told you that with the immunofluorescence, we see when we recruit SIM, we don't see SUMO enrichment. So here, by using the same strategy, you can see SIM is on the left, and in the middle is the telomere, and the telomere is in green on the merged one image on the right. Next, please. When we turn on the dimerization, you can see the dimerizer still works, meaning that the SIM mutant is still recruited to the telomeres. However, you don't see the rounding and the clustering that we see when we recruit SIM. Next, please. You can see from these images as well. Next, please. And when we quantify, you can see SIM, that's the magenta, the integral intensity on the left, the SIM start to increase over time, but it doesn't increase further. And if you look at the telomeres, they also behave differently in that the intensity is not increasing anymore. And in fact, this starts to decrease because of bleaching. And on the right, you can see the quantification of the number of foci. You don't see decrease in the number of telomere, which is in green in this case after we recruit SIM mutant. So this agrees with the hypothesis where the SIM mutant does not lead to enrichment of stromal, does not lead to phase separation. Next, please. And then we want to see if we can reverse this condensation. So if the cell uses simulation to trigger phase separation, then simulation is a reverse process. Then for the cells to reverse telomere clustering, they can simply reverse stimulation. Can we mimic this process then? So what we did then, we incubate the cells with the dimerizer, then allow the telomeres to cluster, allow phase separation to happen, and that is the cells that you see here. We put on the microscope, and then we start to reverse the dimerization as I play the movie. Next, please. You can see as a reverse dimerization sim first is released from the telomeres, but also you can see that the telomeres start to become more dilute. And next, please. If we show you two cases, how that happens is I see cases with the dot starts to dissociate. Meaning, if you look at that bright spot, it becomes two spots. And then on the right, showing you that sometimes one dot simply become more diffusive, meaning that it's like single proteins are leaving that dot. Next, please. And then if we look at the quantification again, look at the intensity of the foci over time, and you can see both channels start to reduce. And the sim is because it's released from the telomere, but the telomere also is reduced. The intensity is reduced because of reversal phase separation and also reversal of the constraint. And if you look at the number of sim is in the magenta, which decreases, but the number of telomeres then actually start to increase because the reversal of telomere constraint. Next, please. So this agrees with hypothesis where stimulation as a DNA damage response on the damaged or stressed telomere then can lead to the ABB formation. And then the fusion of those ABBs, of those liquid ABBs, can lead to telomere clustering. Next, please. Then we next asked that, is this phase separation process can actually create a environment to enrich DNA repair factors? And next, please. So we hypothesized that the driving force for the ABB formation is sumo-sim interaction. Therefore, for proteins contain sumo-sim, that they can be enriched to this ABB condensate. However, we also have a reason to believe this might be more tightly regulated because we know that, for example, stimulation has crosstalk with phosphorylation, which is another response after DNA damage response. Next, please. So to see which one is true, and then we just use immunofluorescence to detect those known ABB components, see whether they are recruited to the de novo ABBs. Next, please. Next, please. Thank you. So here we here is showing you the telomere DNA fish on the left, and we use first look at the PML protein, which is the component, signature component of PML body. And this is a just endogenous cells without recruiting anything. And you can see that the PML protein, we can see some colocalization with the PML protein and the telomeres. And those are, by definition, ABBs. And those are endogenous ABBs that exist in those cancer cells. Next, please. And when we recruit sim, you will see now there are more white dots, meaning more colocalization of PML on the telomere, meaning more number of ABBs. Next, please. But this is not the case when we recruit the same mutant that cannot interact with sumo. Next, please. And those are quantifications showing the difference in either the number and the intensity of the ABBs in those cases, where when we recruit sim, we see a dramatic increase in the number and the intensity of the ABBs. Next, please. But we don't see the enrichment of gene repair factors to the induced ABBs. And we've tested a couple, including P53BP1 and other repair factors. Showing you one is the PCN, which is a cofactor used for the polymerase in those cancer cells. And you can see where the positive control, where we use the Falkland nucleus to induce DNA damage. And you can see PCNA is enriched on the telomere as shown here on the merged image and the number of white foci. Next, please. And when we recruit sim, we don't see such enrichment. Next, please. The same as for sim mutants, so we don't see a lot of PCNA localization to the telomeres after we recruit them. Next, please. Interestingly, then, if we in the after DNA damage, we know that PCNA is modified by sumo, and then if we artificially just fuse PCNA to sumo, in this case to sumo1, we'll see that actually after some improvement, we see PCNA now that when it's fused to sumo, it's highly enriched on the telomere, as you can see the localized white dots on the right. Next, please. And this is not saying we just recruit the same mutant. Next, please. And you can see the difference when we quantify the number of localized foci or the intensity of the localized foci on the right. Next, please. So this suggests that the induced condensates are indeed APBs because they contain the PML body, the PML proteins. However, they're not enriched. The other DNA repair factors are not enriched. But if we artificially allow them to be modified by sumo, then they can be enriched in the condensate. This indicates that this enrichment of the DNA factors, meaning that the composition of this condensate is under tight regulation of DNA damage response. Next, please. So if a model of this kind of true function of the ABB is true, then indicates that the first function should be decoupled from the second function, meaning that the first function only relies on the liquid property of the ABB condensate, and then the second one it depends on its chemical composition. So to test this hypothesis, we want to induce condensates with a different chemistry other than ABBs on the telomeres. So we test the couple shown you one where we use a disordered region RGG from a elastase protein, which is known to phase separate. Next, please. And this is showing you here the images before the dimerization. On the left is the RGG, and in the middle is telomere. Next, please. When we turn on the dimerization, you will see RGG is recruited, but similar to when we recruit them, you can see now RGG start to phase separate because the foci start to round up. Next, please. As you can see in the images, and then again, if we quantify the intensity over time of those foci, you see increase in intensity in both because of the condensation and the clustering. And then the number of foci changes over time in both channels after SIM is recruited, after the RGG is included because of the clustering. Next, please. And so this leads to the conclusion where we show that the simulation is a signal that the cell used to nucleate de novo nucleate ABBs after the damage response on the telomere. It's likely maybe the signal the cells used to dissolve ABBs as well. The reason, the importance for dissolving ABBs is the cells kind of temporarily hold the telomeres together. However, they don't want to hold them for too long. At least they need to dissolve them before the next cell cycle so that they can have accurate cell division. So we think this reversible stimulation process is the cue the cells use to nucleate ABBs, particularly on the telomere after triggering a DNA damage response. And we've also shown that the liquid property of the ABBs is sufficient to drive telomere clustering. And however, the recruitment of DNA repair factors is regulated by other DNA damage signaling. Next, please. So with that, I'd like to thank the lab. Ronwei and Meng contributed to this work. I also want to acknowledge this work is initiated when I was postdoc at Penn with Manson, and it was a collaboration with Hemowit's lab who created the chemicals for us, with the Greenberg lab for the odd cancer. And we are recently funded to study how phase separation organizes the genome of the odd cancer by the 4D Nuclear Consortium, and we are looking for postdocs. So if you're interested in see what is the consequences of phase separation and using tools to actually demonstrate those functions, and please contact me. With that, I want to thank Mark and Joe again for organizing this event, and thank you for having me, and be more than happy to take any questions that you may have.
Thank you so much. Thank you. Wonderful, wonderful lecture.
Thank you.
You covered so much so quickly and clearly. It was really beautiful. It's just beautiful work.
Thank you. Good to hear.
Yeah.
Encouraging words.
Yeah, and you were also very gentle about the next slide, please. that all worked out really well. So I think there's a question from Will Chen. Will, if you want to unmute and ask your question, that'd be great.
Hold on, I'm being a little slow, but I'm going to unmute you, Will.
Okay.
All right, there you go.
Thank you. Hey, really nice talk. Thank you. So for
Thank you.
this concept of upregulating the telomere mechanism I guess it must be for cancer survival or proliferation. Are there any specific tissue types or genomic biomarkers that tell us, is it a pan-cancer mechanism? Is there a specific cancer that depends on this?
So they don't have a specific tissue, but they are very enriched in soft tissue, like nerve system, and also some found in the bone. For example, commonly used U2S, right, bone cancer, and that is OD positive cell line. And sometimes you find they have a subpopulation in some of the cancer cells that use OD. They could exist with cancer cells that use actually telomerase in the same tissue. So,
thank you.
We have a question from, I'm not sure who exactly it is, but Biplab KC. It's a good question, actually. If Joe, you could unmute them.
Hello, I am a student from Japan, so it's midnight here, so I'm sorry I'm not running my video. My question is that—
thank you for tuning in, even though it's so late. I'm flattered.
Yeah, actually, I'm doing research and it's like synthetic system, so I'm quite interested in the biological system of the LLPS system. So finally, the one point that I'm very interesting is that you dissect the two properties. One is the liquid properties and one is the functional properties of the system. And finally, you said that even if you have the liquid property that allow the clustering of the telomerase, the elongation part of the telomerase itself, you are not able to find, right? Because I think it might need some other proteins or other factors from the DNA damage response. So I was wondering, like, what is your next plan then? I mean, even if you cannot then open the telomerase elongation, finally, which is your main target, I think. So what is your next plan? How are you going to tackle this problem? I mean, because the proteins that might be responsible, there might be many proteins, right?
So your question is what the proteins are recruited, or like what? I didn't get really what your question was.
For example, for telomerase elongation, maybe I'm not quite familiar in the field, but maybe there are many proteins are responsible. You need many proteins to be enriched in the liquid phase for the telomerase elongation, right? So, but you cannot find. I mean, you did obtain the pCNA using the fusion protein, but fusion protein for all of the proteins is maybe not feasible practically, I think. So
that's a great question. So there's recent work, not from my lab, but from other labs, showing that when pCNA is downstream, right? So we're just showing that can be enriched or not. It's not going to be trigger the gene damage response. And I think the recent work showing that there's a helicase called BLM, which has the same domain, is required for that protein to be recruited to ABB. And once that protein is recruited, then it can trigger lots of downstream and then initiate the DNA repair.
Okay, so that means there's some checkpoint. Like if you can recruit a single or some proteins only, then it will downstream or other signaling to recruit other proteins.
Yeah, so there's like a series of signaling happening after ABB formation even.
Great. Okay, let's go on to Bede had a question. Thank you. Bede, you're up.
Great talk. Can you hear me?
Yes.
Yeah, I miss you at Penn. So I might have missed this in the talk, but like at the end, you did this LAF1 experiment where you swapped in the LAF1 IDR, which has RGG domains, and those RGG domains can interact with RNA. do you know if generally there's an RNA component to the maintenance of these APB bodies, and is there transcription occurring locally?
That's a good question. There is, but we don't know what is the relationship yet. So there is a telomere RNA that's being transcribed in those cancer cells that is upregulated. And if you knock down the RNA, you will see phenotype. But how the RNA is involved in this process is not super clear yet. One hypothesis, well, there's one study showing that the RNA can form a loop to help this DNA damage response. But also there are other hints that the presence RNA can help maintain a particular chromatin status of the telomere so that the DNA damage response can happen. Which one it is, or that directly linked to APB formation, that is not very clear. But definitely they do play a role by studies showing that if you knock down, they are upregulated, there is upregulation of transcription.
Thanks.
That's great. So let's go to Anirban. That's a really interesting question about heterogeneity. We could unmute Anirban.
Can you hear me?
Yes.
Yes.
Yeah, thank you very much for the insightful talk. I have two quick questions. The heterogeneity, in the first slide you showed that there is a heterogeneity in the formation of this telomere bodies. So how in the cells in which it is not forming, does their condensation is not happening, or what is the phenomena regarding that? And the second question is there is a G quadruplex involvement in telomerase maintenance. So how does that affect in the LLPS phenomena?
Good question. The first one, you're asking the heterogeneity in the telomere length, right?
Right.
So that is an interesting question. We don't know. We think is the reason is the heterogeneity arises from this kind of not well controlled homologous recombination. Compared to telomerase, where it's a really good, well controlled because it's used in stem cells. And so the heterogeneity, but how do cells deal with that? You have extremely long telomeres, you have extremely short telomeres. And we know that when telomeres become very short, they're gonna trigger some response. One of them is actually this start to trigger transcription of the telomeres RNA.
Yes.
So we don't know the shorter ones particularly trigger ABB formation or not. So when we look at ABB formation, endogenous ABB, right? So they are not localized to all the telomeres. Looks like only some of them have it. But are those the ones that are actually very short that trigger damage response, or are the ones that happen to be having a lot of stimulation happen, so you trigger formation? Those we actually don't know.
Okay.
And then for the second question is the G quadruplex. Yes. So the understanding is the telomeres in those cancer cells compared to normal cancer cells, they are in some sort of stress condition, meaning that their telomeres are not well protected and forming those loops, and then they are lacking some proteins. So in this kind of stress condition is when DNA damage response is allowed to happen. But in this kind of stress condition, then you might have more G quadruplex formation. But how does that G quadruplex formation? Either it's G quadruplex of the DNA itself, or actually there is also reason to believe that the G quadruplex can form between DNA and the RNA itself. Then how do cells deal with that? And the kind of current thought is because the presence of the G quadruplex, therefore those telomeres, they have more trouble replicating, and that leads to replication stress. And that replication stress leads to DNA response, and then it becomes a loop allow those cancer cells to kind of survive under those stress conditions, because the stress allows them to survive. And you know what I'm talking about? Like the whole,
Right.
like, yeah.
Like it's a feedback loop. It's a feedback.
Yep, it's a feedback loop where the stress is there, but then it's the stress that allows them to survive.
So that means in a stress condition, say in a under chemotherapy, so cells which have these bodies may survive, like due to the heterogeneity, we know that it greatly affects the chemoresistance property of cancer. So once chemotherapy is inducing these DNA damages, or a stress condition, means it is selecting the cells which are having these particular bodies. So is there any chance of that happening?
Yeah, there's some evidence believing that the stem cancer cells particularly like to use ART pathway because they are so stressed. They allow them to survive. Also, I mentioned earlier on that when telomerase positive cancer cells under stress conditions, under the telomerase inhibitors, then some of them will not die and then adopt ART pathway and then to escape that inhibition. And this is actually a huge problem for the cancer therapy for telomerase inhibitors.
Yes, and like in the recent work, like these chemotherapy drugs can partition into the condensates. So it might be interesting that if these commonly used drugs is getting partitioned into these condensate bodies at the telomeres.
That's a great thought. Yes, I would imagine, like, for the ones that particularly enrich in the PML body would be very interesting to try with all cancers, right? But again, in general, right, targeting telomeres has this kind of drawback with it's slow. So probably some sort of combination will be ideal where you kill rapidly with other drugs like microtubule drugs, but then you inhibit the ART pathway so they don't become like stem cancer cells. I think that will be very interesting.
That makes a lot of sense.
Thank you.
Thanks, yeah. Thanks.
Thank you.
And so building some more on PML, Will had another couple of questions relating to that.
Hey, Huaiying, one question I had was, like Mark is saying that, so the reason I asked about whether there's a stratification of the telomere mechanism is that there, in general, DNA damage and chromosomal instability features in different kinds of cancer, subtypes of cancers. So this mechanism that you have of recruiting PML bodies, do you see any evidence or have you any ideas for whether that could be harnessed in other repair situations by the cancers?
PML protein itself and PML bodies are implicated in DNA damage response. Particularly, I think a lot of work early on showing PML body is a sensor for DNA damage. And there are also some work showing that PML protein forming this they call nanobody that locally kind of form at the DNA damage response to help with the DNA damage response. So it could be, but I don't think it's clear how it's involved. For example, in homologous recombination, does it involve in recruiting which protein? I don't think that is clearly demonstrated, but there's definitely correlation or actually co-localization of PML nanobody to the damage sites. And there are more co-localization when cells actually fail to actually repair DNA damage. So you will see those kind of persist DNA damage foci, then goes to the PML body, or I don't know if the nuclear PML body, or they actually just go to the PML body because they move around and then they start to fuse. I think at least I think it's comfortable to say that the PML body is linked with the DNA damage response, but I don't know if it's a kind of direct therapy. You could use if by using the power of PML as body as a sensor for DNA damage, then you could kind of inhibit DNA damage response. For example, there are some drugs where you can inhibit DNA damage response, and if you also inhibit transcription, and then that seems to be actually better for the therapy. So they think of a sort of a combination, meaning because of the the cross-modification transcription and the DNA damage response and the PML body. So from that point of view, I can see that it might be reasonable. And also in general, PML body is a stress responder, right? So for overexpression, for heat stress, for example, even those kind of stresses, PML body is involved in it. But again, what is the particular role of PML body? I don't think it's super clearly defined as a molecular pathway.
Excellent, thank you.
That's a great answer. It's a complicated topic. It's not going to go away. That's my prediction. Is we'll keep learning more about this. So maybe we're at a good stopping point. We've run a little bit over the hour. There's some great questions, wonderful answers, wonderful lecture. So
Thank you.
This has been fantastic. You know, Huaiying Zhang is a amazing speaker. Clearly, we've all learned that. You know, and doing wonderful research. So thank you for your time and thank you for doing this for the whole community.
Thank you for the opportunity.
Sure. All right. Take care, everyone. Thanks for joining us.
Thanks.
Bye.
Thank you.
Bye-bye.